Hello there, ladies and gentlemen. Mr. Williams here, and welcome to your unit over complex patterns of inheritance. Big picture ideas here for this unit as we go forward: heredity and genetics. Heredity being the passing on of information from parent to offspring, and genetics being kind of the field of study that really analyzes that passing of traits and the mechanisms that control the passing of traits from parent to offspring. We're going to set some foundational information here in this video. We're going to set a good foundation for us as we go forward, and we're actually going to cover one of those mechanisms that deals with kind of the organization of traits prior to the passing on. Okay? We're going to look at the formation of sex cells. We're going to talk about meiosis. That'll be the first video topic that we look at. Sounds a lot like mitosis, and it is, but we have some major differences that we'll cover. Second video topic is going to deal with the genetic variation that is established through meiotic division. Okay, video topic number one, meiosis. Meiosis is a process that gives us gametes, or sex cells, that are used in eukaryotic organisms when they are sexually reproducing. It is what we call a reduction division, and its name is very close to mitosis. We are making more cells when we go through the meiotic process, meiosis, but we're also reducing a certain item, and that certain item is the amount of genetic information that is present in the product. Okay? So we're making more cells with meiosis, but we're actually cutting in half the original or typical amount of genetic information that was originally present. Okay? So it's a reduction division that gives us gametes or sex cells. It's really, really close to mitosis in regards to the movement of chromosomes throughout the entire process. Okay? So we have the same phases, and we pretty much have the same mechanisms at play here. But there are some major differences that we'll get into here in a second when we look at our simulation. Those major differences being there are two nuclear divisions throughout the whole meiotic process. We've also got two genetic variation increasing mechanisms known as crossing over and independent assortment taking place. And finally, the last major difference, there is no DNA replication going on between the two nuclear divisions. No DNA replication taking place, hence giving us the reduction and the final amount of genetic information in each one of the resulting cells. Let's take a look at that simulation here. Okay, here we are with our diploid germ cell that's getting ready to undergo meiosis. We enter prophase 1 of meiosis 1, so the first phase of stage 1 of meiosis. We see some similarities between meiosis and mitosis here in prophase 1. We have the condensation of chromosomes, okay, or the condensation of DNA into chromosomes, excuse me. And we've also got the forming of homologous chromosomes. But there's something special going on here. We've got these homologous chromosomes forming homologous pairs. And you start to see this here as the nuclear envelope is dissolving and the spindle apparatus is moving into place, okay, along with the spindle fibers. What we see in prophase 1 is the forming of homologous pairs between two homologous chromosomes. Okay? For instance, the blue and red homologous chromosomes have formed a homologous pair, and the purple and yellow homologous chromosomes have formed a homologous pair. Now, one of the unique mechanisms that's taking place here in prophase 1 of meiosis is crossing over. And it's going to increase the genetic variation of the resulting sex cells greatly. We've actually got like segments of DNA within these homologous chromosomes swapping between each other. So you can see that we've got crossing over happening between our blue and red homologous chromosomes and between our purple and yellow homologous chromosomes, giving us some genetic variation within the homologous pair. Prophase 1 blends into metaphase 1, and just like metaphase 1 of mitosis, we've got lining up on the equator and the attachment of the spindle fibers to the homologous pairs. Now, the homologous pairs through anaphase 1 are going to separate, and they are going to be pulled to their respective poles or areas of the initial cell that's undergoing this first nuclear division. We get the telophase 1, and we may or may not have the reforming of a nucleus. It just depends kind of on the organism that we are studying. Telophase 1 blends into prophase 2. And notice, between meiosis 1 and meiosis 2, those two stages, no DNA replication. No DNA replication. Okay? So these homologous chromosomes are now going to reline on the equator in metaphase 2. The spindle fibers are going to reattach to the centromeres of the homologous chromosomes, and through anaphase 2, the homologous chromosomes are now going to be split into their sister chromatids, or back to the term we use, chromosome, okay, the actual chromosome structure itself, and they are going to be pulled to the respective poles of their cells. And notice now we've got 4 nuclei present. Cytokinesis is taking place between the original large cell and the 2 newly created cells from the original gigantic cell, and the final product, 2 sets of haploid sex cells. So a total of 4 gametes have been created, each one with half the original amount of genetic information that diploid cell would be possessing during the regular interphase stages of its life. Now, one of the genetic variation mechanisms that I did not mention was independent assortment. And independent assortment is essentially referring to the lining up, the random lining up of the homologous pairs and homologous chromosomes on the equator, and the separation and movement, entire random lining up and separation and movement of the homologous pairs and homologous chromosomes on the equator to the separate sides of the poles. So you can see here that we've got the red chromosome of the homologous chromosome at the top here in metaphase 2. It's going to go to the left of that top cell, whereas the red and blue homologous chromosome is going to go to the right. That could be flip-flopped. Okay, that could be flip-flopped. It could have lined up differently upon its entering into metaphase 2. Same thing with the homologous chromosome below it, the yellow and then the yellow with the little purple towards the top. It could have lined up maybe towards the top and flipped around to where the yellow and purple would have gone to the left and not to the right. So independent assortment refers to the absolute random alignment of these homologous pairs during metaphase 1 and homologous chromosomes during metaphase 2, and their random separation during anaphase 1 and anaphase 2. Increases the genetic variation. Independent assortment. Very important process. Okay, to get another angle at meiosis and some historical information and background, we're going to check out a little clip here from Mr. Bill Nye.
Around the time that Walter Fleming was studying the cell division process, biologists already knew that fertilization resulted from the union of 2 sex cells, sperm and egg. What they didn't know was why these were the only types of cells in the body capable of reproducing new life. What made them different? The first answer was provided in 1883 by Belgian zoologist Edward van Beneden. Van Beneden was studying a species of roundworm when he discovered that nearly all of its cells contained 4 chromosomes. The only exceptions were the roundworm's sex cells, the male sperm and the female egg. These cells had only 2 chromosomes. Van Beneden observed that once 2 of the sex cells came together, a full set of 4 chromosomes appeared in the fertilized egg. But just why the sex cells had only half the number of chromosomes compared to all the other cells was still a mystery. Then in 1887, German biologist August Weismann entered the picture.
He knew about the work of Van Beneden, who had shown that the sperm and the egg bring in equal numbers of chromosomes. But Van Beneden's work had not really worked out the details of how the chromosomes were reduced in number.
He saw them go in half, and he saw them recombine.
He saw that the sperm and the egg brought in half the number of chromosomes as the body cell, but he didn't really know how they got rid of the half that they got rid of. So Weismann discussed how this might happen from a theoretical standpoint. And he said, well, what you could have would be that either every one of the chromosomes would just split in half, and half would go off one way and half would go another way, or you could have it more complicated, that the chromosomes could pair, and then instead of splitting, they just separate again.
After years of studying the behavior of chromosomes, Weissman determined that at some point a developing organism signals its sex cells to divide their number of chromosomes in half. Weissman called this very special form of cell division meiosis.
I think they will look back and see this as the golden age of biology, because, let's say, 200 years, between 1800 and 2000, we went from not knowing what distinguished a stone from a frog. Okay? And now we know the difference between a stone and a frog.
That's pretty good.
Yeah. So that, in a real sense, we've answered the old philosophical question of what is life. Maybe not to everybody's satisfaction, but we're well on the way.
Thank you, Mr. Bill Nye, for that. We appreciate it. And that does it for meiosis here. Okay, in review, we should feel comfortable with meiosis being a reduction division that gives us 4 haploid gametes, 4 haploid sex cells. Very similar to mitosis, okay, except there's 8 phases, all right? 2 stages, meiosis 1, meiosis 2, all right? 2 nuclear divisions going on. No DNA replication happening between the 2 nuclear divisions, all right? And we've got 2 mechanisms that are increasing the genetic variation for those resulting sex cells, those 2 being crossing over and independent assortment. That's all I have for you in this video. Thanks for tuning in. If you have any questions, ask your teacher. See you guys later.